So in 1942, the British military did this highly classified test on Grunard Island. This is a tiny, windy little piece of land off the coast of Scotland. And they dropped bombs on it.
Right, but not traditional explosives. You know, they didn't blow things up.
Exactly. They released this fine, completely invisible biological powder. And the crazy thing is, for the next 48 years, that island was considered so dangerous that just setting foot on it meant almost certain death.
Yeah, it wasn't until, I think, 1990 that they finally declared it safe. And they had to soak the entire island's topsoil in formaldehyde to do it.
Which is just wild. Now, the organism they dropped was Bacillus anthracis, the bacterium that causes anthrax.
But here's the thing. It wasn't the active bacteria that kept the island lethal for half a century. I mean, active bacteria would have just died off in that harsh Scottish weather in a matter of days.
Right, so why was it still deadly? Because those bacteria deployed a sort of microscopic survival mechanism. They basically retreated into an armored state that, well, it just completely ignores the passage of time.
Yeah, they just wait.
And welcome to today's deep dive. We're sorting through a stack of microbiological sources to understand exactly the endospore. Our mission today is to figure out how certain bacteria build these invisible, practically indestructible survival capsules.
And why this phenomenon, the exact same one that shut down an entire island for decades, is actually dictating the safety of things in your daily life, like the canned food in your pantry or the soap dispensers in your local hospital.
Okay, let's unpack this. Because to really understand the endospore, we first have to, totally throw out everything we assume the word spore actually means.
Right, because the term is kind of a biological false friend. I mean, when you hear spore, your mind probably immediately goes to fungi, right?
Oh, totally. Like a mushroom in a forest or mold on a piece of bread.
Exactly. Fungi use spores to reproduce. A single fungus will generate millions of spores, release them into the wind, and just multiply to conquer new territory.
But an endospore is the exact opposite of that. It's not about reproduction at all.
No, it's the absolute antithesis of reproduction. It's just a sheer, desperate act of self-preservation.
Yeah, the math laid out in the research really highlights how desperate it is. Like if you're looking at a normal, actively dividing bacterial cell, what microbiologists call a vegetative cell, and its environment suddenly turns hostile, it doesn't try to multiply.
Right. It triggers a process called sporulation, and the final tally of that process is exactly one endospore.
One vegetative cell creates one spore. So zero population growth.
Zero. And it's worth noting this isn't some universal bacterial trick. A standard gram-negative bacterium like E. coli or Salmonella, if things get too harsh, they just die. They literally lack the genetic software for this.
So who can actually do this?
It's almost entirely restricted to this specific slice of the bacterial world, specifically gram-positive bacteria in a phylum called Firmicutes.
Okay.
The most common ones you'll run into are in two genera. There's Bacillus, which needs oxygen to live, and Clostridium or Clostridioids, which are anaerobic, meaning they only thrive in oxygen-free environments.
I really want to walk through the actual construction of this thing, because it's not like the cell just spontaneously hardens. It senses starvation or extreme heat or toxic chemicals, right?
Yeah, it realizes it has no immune system to fight back, so it goes into full biological lockdown.
The sources describe the trigger as a phosphorylation cascade, which is, well, it's basically a microscopic game of telephone inside the cell.
That's a great way to put it.
Proteins just start frantically tagging each other with energy molecules, passing this panic message down the line until it reaches the DNA. And that activates a master regulator protein called SPO-AA.
And SPO-A is the point of no return. Once that's activated, normal division stops. Usually a cell replicates its DNA and splits perfectly down the middle, right? Two identical daughter cells.
Right.
But under SPO-A, it does an asymmetric division and makes this small compartment at one end called the forespore and leaves a much larger compartment called the mother cell.
And then the mother cell does something just incredible. It literally swallows the forespore.
Yeah, engulfs it entirely.
It wraps that smaller compartment in a second membrane. And from that moment on, the mother cell dedicates every last drop of its energy to building layers of armor around the part it swallowed.
It's totally, completely selfless.
And when the construction is finally done, the mother cell bursts open, it leases, and it dies, leaving only the fully armored spore behind. It's like building an indestructible escape pod, locking your DNA inside and then blowing up the mothership to launch it.
Exactly. The mother cell just becomes a temporary scaffolding, and a fuel source to manufacture those defenses.
But wait, a cell just dropping a second membrane over its DNA that doesn't explain how it survives boiling water or chemical attacks.
Right.
I mean, if you drop a normal bacterial cell with 2 membranes into boiling water, it still cooks.
Oh, absolutely.
So is it just a really thick physical wall, or is there some kind of chemical wizardry happening inside?
What's fascinating here is that the internal chemistry is completely overhauled, and it starts with removing the most essential ingredient for life. Right. The primary defense mechanism of an endospore is extreme dehydration. The core of the spore, where the DNA is, has almost all of its water physically pumped out.
Which, if you think about how physical damage happens at a microscopic level, makes perfect sense. Water is basically the medium that facilitates destruction.
Yeah, if you want to denature a protein with heat effectively cooking it, you need water to break and reform those chemical bonds.
And with radiation too, right? If radiation hits a cell, it usually hits water molecules, shattering them into free radicals that shred the DNA. By taking out the water, the spore freezes its chemistry in place.
The destructive reactions literally have no medium to travel through. But to achieve that crazy state of dehydration, the spore packs its core with a unique compound called calcium dipicolinic acid, or KDPA.
KDPA.
Yeah, and it can make up a massive percentage of the spore's dry weight.
It functions almost exactly like biological antifreeze. Like you swap out water for coolant in a car radiator before winter so the engine block doesn't crack, right?
Exactly.
The spore replaces its internal water with KDPA. It binds up any remaining moisture and physically wedges itself into the DNA helix, locking the genetic code in place so it won't unravel when it gets hot.
And the DNA gets a second layer of direct protection, too. It's tightly coated by molecules called small acid-soluble proteins, or SIPEPOS.
Hey, Postbase, I was reading the molecular diagrams for these, and they're brilliant. They act like a cross between microscopic bubble wrap and origami.
Origami is a perfect analogy.
Right. They latch onto the DNA strands and physically twist them into a tighter, more compact shape.
Yeah, by altering the geometry of the DNA, the ASPs tuck the most vulnerable chemical bonds away on the inside. So things like UV radiation from the sun just can't access the sensitive parts of the code to cause fatal mutations.
Okay, so we've got a totally desiccated core, stabilized by KDPA antifreeze, and
Well-
shielded by SASP bubble wrap.
Right, but you can't maintain a desiccated core in a wet environment, like soil or a human body, without a heavy-duty container.
Yeah.
So surrounding the core is this thick layer called the peptidoglycan cortex. It functions like a rigid pressure vessel. But instead of keeping pressure in, it mechanically resists the osmotic pressure from the outside world, refusing to let water flow back into the dry core.
And outside of that, you have multiple dense layers of protein coats. They act as a physical sieve and a chemical shield, blocking enzymes or harsh chemicals.
And in some species like the Bacillus anthracis on Grunard Island, there's an additional balloon-like outer layer called an exosporium.
And that's designed to help it evade the immune system if it gets inhaled by a host, right?
Exactly. So what you're looking at is a nested architecture of survival. Exosporium, protein coats, rigid cortex, KDPA antifreeze, and SASP wrap DNA.
But here is my problem with this whole design. We've built the ultimate microscopic tomb. It's totally dehydrated, heavily armored, and I mean, textbooks have historically classified these spores as having zero detectable metabolism.
Not at all.
Right, they don't eat, they don't breathe, they produce zero energy. So wait, if textbooks say these spores have zero metabolism, how on earth is a quote-unquote dead thing sensing its environment to wake up? A panic room is totally useless if you can't hear the all-clear signal, you know.
That's the real magic of it. The awakening process germination relies on a beautifully simple mechanism that requires no active energy from the spore itself.
None.
None. Woven into the spore's inner membrane are these dedicated passive receptor proteins. They just sit there completely inert, acting as microscopic tripwires.
Just waiting.
Just waiting for the physical shape of a specific germinant molecule to bump into them. Often it's something as abundant as a single nutrient, like the amino acid L-alanine.
So the nutrient just slots into the receptor like a key falling into a lock by pure chance.
Exactly. And the moment that molecule binds, it trips a structural change that initiates an irreversible chain reaction. The spore immediately dumps all of its calcium DPA out into the environment.
And without the KDPA, the water just rushes back into the core.
Yep. Specialized enzymes that were completely dormant suddenly activate and start chewing through the rigid cortex from the inside out. Within minutes to a couple of hours, the armor dissolves, it rehydrates, and it emerges as a normal, actively growing cell.
Ready to multiply.
Yeah.
So what does this all mean? The fact that they can flip from a heavily armored, lifeless state back into a growing pathogen in a matter of minutes that dictates the safety protocols of our entire modern world. So let's pull this out of the microscope and into your kitchen.
Okay, yeah.
Because if you do any home canning, you are dealing directly with endospores.
You definitely are. The major threat in food preservation is Clostridium botulinum. It produces botulinum toxin, literally one of the most lethal neurotoxins on the planet. And crucially, it forms endospores.
And since Clostridium is an anaerobe, it only thrives where there is no oxygen. So when you preserve low-acid foods at home, like canning green beans or carrots, you seal them in a glass jar and drive out the air. You're custom building the ultimate oxygen-free real estate for C. botulinum.
And a standard practice for many people is to just boil those jars to sterilize the food inside. But boiling water on a stove maxes out at 100 degrees Celsius, right, 212 Fahrenheit.
Right.
That temp will easily destroy the active bacteria, but it does absolutely nothing to the C. botulinum endospores. The CSPs and that dehydrated core just shrug off 100 degrees.
Wow. So the spores just sit in the boiling water, fully protected. You pull the jar out, it cools down on your counter. And the spores sense the nutrients in the green beans. They trip that receptor, shed their armor, and start actively growing in the low oxygen vacuum of your jar, pumping lethal botulinum toxin into your food.
Because boiling does not equal sterilizing.
Exactly. This is the exact reason food safety guidelines absolutely mandate the use of a pressure canner for low-acid foods.
Yeah, a pressure canner traps steam, driving the internal temperature up to 121 degrees Celsius, 250 Fahrenheit. The combination of that extreme heat and the intense physical pressure forces thermal energy straight through the protein coats and the cortex,
ultimately denaturing the SASPs and cooking the spore from the inside out.
And failing to use a pressure canner is why we still see fatal botulism outbreaks tied to homemade preserves today.
It's terrifying. And this invisible armor also forces hospitals to totally change how they operate, especially regarding hand hygiene. Like, we've all been conditioned to believe that a quick pump of hand sanitizer is the ultimate shield against germs, right?
Oh, yeah. And alcohol is incredibly effective against vegetative bacteria and many viruses because it dissolves their lipid membranes. But an endospore's outer layers are made of highly cross-linked protein coats, not a vulnerable lipid bilayer.
So the alcohol physically cannot penetrate the shell. The spore just ignores it.
Completely ignores it. This becomes a really terrifying reality in hospitals dealing with Clostridioides difficile, or C. diff, which causes severe, highly contagious gastrointestinal infections.
When a C. diff outbreak hits a hospital ward, the protocols change instantly. Alcohol sanitizers are basically banned.
Yeah, medical staff are required to wash their hands strictly with soap and water.
But the soap doesn't actually kill the C. diff endospore either, right?
No, it doesn't kill it. But the surfactant properties of the soap, combined with the physical friction of scrubbing, break the spore's grip on the skin. You're literally washing them down the drain.
And for sanitizing hospital surfaces during an outbreak, alcohol wipes are useless. Cleaning crews have to switch to harsh, highly concentrated bleach disinfectants.
Because bleach acts as a powerful oxidizing agent that can chemically dismantle those tough outer spore coats over time.
Hospitals actually lean into the sheer toughness of spores to guarantee patient safety too. Like with autoclaves, those industrial pressure cookers used to sterilize surgical instruments. They regularly test them using biological indicators.
Yeah, they intentionally put vials of endospores inside the autoclave right alongside the surgical tools. And not just any spore, they use a specific benchmark organism called Bacillus stereothermophilus.
Which is brilliant logic, honestly. You don't test your sterilization equipment against an average pathogen. You test it against the absolute heavyweight champion of heat resistance.
Right. If the autoclave cycle finishes and tests confirm the Bacillus spores are completely destroyed, you have mathematical certainty that every other lesser pathogen on those scalpels is dead too.
Our ability to safely perform surgery or preserve food today, it all stems directly from the fact that 19th century scientists were driven absolutely mad by these organisms.
Oh, they really were. In the 1870s, the scientific community was embroiled in this massive debate over spontaneous generation, the idea that life could just magically appear out of non-living matter.
And this physicist, John Tyndall, was trying to definitively disprove this. He would take a flask of nutrient broth, boil it to sterilize it, and seal it. But a few days later, the broth would often turn cloudy, just teeming with bacteria.
And his critics pointed to this as absolute proof, life spontaneously generating from nothing.
But Tyndall was baffled until he read the work of a botanist named Ferdinand Kohn. Cohen had been observing certain bacteria under a microscope and noticed these highly refractive, glowing structures inside the cells that actually survived boiling.
He had discovered the endospore.
Yeah. So Tyndall realized the bacteria weren't magically appearing from nothing. They were just waking up. The boiling had killed the active cells, but the invisible spores had survived in the broth. So he invented a method to outsmart them, a process we still call Tyndallization.
It's basically a masterclass in exploiting the spore's own tripwire mechanism.
Tyndall would boil the broth once, killing all the active bacteria. Then he'd let the broth sit at room temperature for 24 hours. The surviving spores, sensing the safe, nutrient-rich environment, would germinate.
Right, they'd shed their indestructible armor to become vulnerable vegetative cells again.
Exactly. And the next day, Tyndall would boil the broth again, killing this newly awakened batch. He repeated this cycle over several days, systematically tricking the spores into disarming themselves and catching every last one.
Which finally proved that spontaneous generation was a myth. And around the exact same time, Robert Koch used Cohen's spore concept to solve the terrifying mystery of anthrax.
Right, because farmers were losing cattle to anthrax in pastures where no infected animal had grazed for years.
Yeah, and Koch proved that Bacillus anthracis formed endospores. The bacteria weren't spontaneously appearing in the cattle. The spores were just sitting dormant in the dirt, fully armored, waiting for years to be ingested by a new host.
If we connect this to the bigger picture, the longevity that Koch observed in the soil is the exact trait that militaries weaponized in the 20th century. By cultivating endospores, dehydrating them into a fine powder, and aerosolizing them, bioweapons programs bypass the normal fragility of biology entirely.
Which brings us right back to Grignard Island in 1942. The British military didn't just contaminate the soil, they proved that an aerosolized spore could withstand the massive environmental shock of an explosion, settle into the dirt, and remain viable for 48 years.
We saw the same grim reality during the tragic 1979 Sverdlovsk incident in the Soviet Union. An accidental release of anthrax spores from a military facility drifted over the surrounding area, causing a massive, deadly outbreak.
And in the 2001 anthrax letter attacks in the United States, spores were mailed in paper envelopes. They sat totally inert in dry powder form, harmless to the touch, but lethal the moment they were inhaled into the warm, nutrient-rich environment of a human lung.
The sheer refusal of these things to die raises a huge question. I mean, we know they can survive half a century in the dirt. But what is the absolute limit of their dormancy? We're moving from concrete human history to deep time here. Some sources pointed to a highly publicized 1995 study claiming scientists revived a bacillus spore from the gut of a bee that was trapped in fossilized amber.
That would mean the spore survived for tens of millions of years.
Yeah. And other studies have claimed to revive spores trapped inside ancient salt crystals deep underground.
This raises an important question, though. Those claims definitely generate incredible headlines, but they face intense skepticism from the broader microbiology community.
Really? Why?
The skepticism comes down to the fundamental laws of physics. Even in a perfectly dormant state, with a dehydrated core and SAST proteins protecting the code, the spore cannot escape background radiation and thermodynamic decay.
Oh, right. Because over millions of years, background radiation from the Earth itself will cause double-strand breaks in the DNA. And because the spore is dormant, it has no water and no active metabolism. It can't repair those breaks.
Exactly. The damage simply accumulates. Over geological timescales, the genetic blueprint should be shattered into just illegible fragments. The mathematical calculations regarding the half-life of DNA simply do not support survival over tens of millions of years.
So how do they explain the bacteria in the amber or the salt?
The far more grounded explanation for those discoveries is modern laboratory contamination. Endospores are ubiquitous. They're in the dust circulating in the lab, on the reagents, on the researchers' equipment, proving definitively that the bacterium growing in your petri dish woke up from a million-year sleep in amber and didn't just float down from the air conditioning vent. It's nearly impossible.
Okay, so millions of years might be a stretch due to radiation decay. But even if deep time is off the table, their physical limits are astronomical.
Yeah.
I mean, the European Space Agency literally took bacterial spores, strapped them to the outside of the International Space Station, and left them there.
Yeah, they subjected them directly to the vacuum of space, massive temperature extremes, and unfiltered cosmic ultraviolet radiation.
And a significant percentage of them survived, especially if they were clustered together, where the outer dead spores shielded the inner ones, or if they were mixed with a tiny bit of meteorite dust.
Which keeps the hypothesis of panspermia scientifically viable, the idea that the building blocks of life, or simple organisms themselves, could survive an interplanetary journey hitching a ride on an asteroid.
What is truly staggering about those space station experiments is the realization of evolutionary overlap. These bacteria did not evolve to survive the vacuum of space. They evolved to survive a hot, dry day in the dirt on Earth.
But the chemical and physical solutions they developed, the extreme dehydration, the CAD-DPA antifreeze, the DNA origami, happen to be the exact same traits required to survive the cosmos.
It's mind-blowing. Before we wrap up, let's just quickly distill this deep dive into the vital takeaways for you. First, an endospore is not a tool for reproduction. It is the ultimate survival bunker, built by sacrificing the mother cell.
Second, it survives through a complex stack of physical and chemical armor, relying on dehydration and specialized proteins to freeze its internal chemistry.
Third, because of this intense armor, normal boiling water is just not enough to sterilize food or equipment. That is why we use pressure canners at home and benchmark stores to test hospital autoclaves.
And the final concept to consider is this idea of superdormancy. We have an organism that can sit in the soil of a Scottish island for 50 years or float in the frozen vacuum of space, displaying absolute zero active metabolism.
Right, it doesn't eat, grow, or breathe. For all intents and purposes, it is inanimate chemistry.
Yet the moment a single molecule of L-alanine bumps into a receptor on its membrane, that inanimate chemistry springs back into complex biological action. It forces us to ask a fundamental philosophical question: does our standard definition of life need to be rewritten?
When an endospore is in that perfectly dormant state, is it a living organism waiting inside a bunker? Or is the bunker itself a paused state of life?
It's wild to think about.
That is an incredible thought to leave you with. Thank you for joining us on this deep dive into the invisible survival capsules hiding in our pantries and hospitals. Keep exploring, stay curious, and we'll catch you next time.